Hi everyone. Thank you for taking the time to join me today for this brief presentation. My name is Peter Fung. I'm the Senior Manager of Customer Marketing at Nanotemper Technologies, and I'd like to discuss with you today how you can tackle small molecule drug discovery screening challenges by using Dianthus. To start things off, I just wanted to address the inefficiencies in drug discovery as they're an ongoing challenge that scientists face. Despite the fact that more money is spent to help support R&D efforts, particularly in pharma, the actual number of new drugs approved by the FDA still remains very low. Some of the causes of this can really be that there are higher regulatory hurdles that scientists need to meet in order to launch a drug product. On top of this, diseases are getting more increasingly complex and harder to study. And along these same lines, it's that researchers don't have the right tools that are able to get them their answers much faster. So how can you really address these inefficiencies or these trends that researchers are facing as they're challenged in drug discovery? And one approach is really to focus on novel approaches and developing new technologies and innovations that help them identify and find the best candidates possible. Some of the more recent innovations that have come out include CRISPR, or gene editing, which has really changed and revolutionized science. There's been a lot of focus on biologics, particularly monoclonal antibodies, ADCs, and biosimilars as being popular targets to help treat patients. Also, there's been a lot of advances in stem cell research that have also shown to be very promising. And more recently, both immunotherapy and gene therapy are techniques that continue to show promise as being able to develop new therapeutics for patients. One class, which I'd like to focus on more today, is small molecules and fragment-based targets, as they really are the mainstay of drug discovery, as small molecules have contributed to approximately 75% of the new drug approvals in the past 5 years. And they offer a unique ability to be taken up by the cells in an easier way than some of the other biologics that are being developed. So I'd like to challenge you all to think about the fact that to achieve more success in drug discovery really requires a focused effort on screening and identifying your best targets. And there are 3 areas that we can improve upon. The first is hit identification, where you find your true hits faster and then move them onto your hit validation more confidently. The second phase, hit validation, is having the ability to detect a wider array of both strong and weak binders as you're trying to characterize and identify your best target candidates. And you need to be able to quickly interpret these results and get your answers. And then finally move them on to lead optimization, where you can verify their binding abilities and then see if you can if the binding affinities remain strong while you improve upon their specificity, selectivity, and potency, as this will really lead you to developing the best drug candidates. So with that, I'd like to introduce you to Dianthus, which delivers fast, non-stop, and highly sensitive hit screening. With Dianthus, you can swiftly analyze 384 data points in just 30 minutes. You can measure a wide range of affinities from picomolar to millimolar KDs. It consumes a tiny amount of sample, both the target and the compounds that are used to be assayed. And all characterization is done in solution, so there's no immobilization required to obtain your binding affinities, which are commonly occurring with some technologies that are available today. And its required process. With Dianthus, it seamlessly fits into your screening and characterization workflows from as early as assay development, where you're just trying to do the initial characterization of your targets, To be able to do single dose screening so you can very quickly screen very large libraries, get your identified hits from those, and move them on to perform a more detailed affinity screen where you can rank order your different targets as well as validate and quantify them. And then finally move them on to the lead optimization process where you can work with your chemists and biologists to do chemical modifications of those lead target hits and increase their binding affinities. To date, our users of Dianthus have screened a wide array of targets, including small molecules, which I'll talk about more today, peptides, aptamers, and fragment-based libraries. Our users have also looked at DNA-encoded libraries as well as many RNA therapeutics. Our Dianthus users have also used the system to examine PROTACs, various enzymes, and a series of different kinases. And of course, many of them are using them to look at biologics, particularly monoclonal antibodies, ADCs, and biosimilars, as well as other large protein complexes, including membrane proteins and GPCRs, to look at protein-protein interactions. But with Dianthus, you can do a lot more than just screening. You can characterize binding events to understand the relationship between molecular interactions and target functions. You can also use Dianthus to characterize cell signaling pathways by knowing how molecules interact with each other that turn them on or turn them off. And also you can use Dianthus to confirm structure-activity relations. This allows you to support and confirm your X-ray crystallography and cryo-EM results. So there's really a broad applicability of using Dianthus. It's easy to use. Anyone in the lab can use it without any specialized expertise. And the assay setup is very flexible and easy to perform. So let's talk more about the technology that's powering Dianthus. Dianthus quantifies molecular interactions using our proven technology called TRIC, which is temperature-related intensity change. With TRIC, the steps involved are that you label your protein with a fluorescent dye, incubate it with a binding partner. You then put it in the system where you apply a precise and laser-induced temperature change that results in changes to the fluorescence intensity that's measured. The TRIC signal is affected by the fluorophore's microenvironment, which can change upon molecular binding or by conformational changes. So to summarize this, binding events are detected by measuring changes in the fluorescence intensity. You have your labeled target, which either has interacted with its binding partner or you have it in its unbound state. The IR laser is turned on briefly to change the temperature of the sample, and as you can see, the unbound state targeted molecule provides a fluorescence signal that's different from that obtained when there's an interaction that occurs, and that's what we're measuring with TRIC, is this change in the signal amplitude. There are many options to choose for different models of Dianthus that are right for your specific research needs. The NT23 is our workhorse system. It gives you nanomolar sensitivity and fast results. Now, if you require more sensitivity, the NT23Pico is super sensitive, delivering picomolar sensitivity. And our NT23PicoDuo gives you the fastest time to results with the highest picomolar sensitivity. And I should mention that all of our systems are amenable to automation for high-throughput screening needs. Dianthus is flexible and fits into any type of research need, including screening facilities where they're looking at a lot of fragment-based drug discovery or screening molecules, or small molecule screening, and various biotechs where they're interested in characterizing biologics and their workflows, including monoclonal antibodies and ADCs or nanobodies. CROs, which provide an important service for screening different samples, Dianthus has become a valuable tool for them. as well as core facilities, which are looking at biophysical and structural characterization, as well as biomolecular structure and functional studies that are shared amongst multiple labs. And of course, both large and small pharma are using Dianthus to screen for drug candidates. So let me show you one example of how Dianthus is used and the data that's generated with it. In this particular example, we're looking at fragment screening against G9A as a target. G9A, for those of you who don't know, is a methyltransferase that methylates lysine residue 9 on histone H3 and has been shown to be involved in epigenetic gene regulation. G9A has also been linked to several diseases including breast and lung cancer and has therefore become an important target to study in drug discovery. Now in this example that I'll show you, this is work done in collaboration with scientists at Imperial College in London, where they used Dianthus to screen almost 2,500 fragments against G9A, covering a broad chemical space in both mass and complexity. Shown by this first step, a buffer screening was performed for G9A. As I mentioned, our target, G9A, and its ligand or control ligand that we used in this experiment was S-odenosyl methionine, or SAM. What's shown on the right is a heat map plot with the results of screening our nanotemper buffer exploration kit, which contains 96 different buffers that are commonly used for studying molecular interactions. A reference was used, which was DMSO alone, and the positive control was the molecule SAM. As seen here in this study, calcium chloride is a good buffer to use, providing very high signals, as shown by these two examples here, that produce a nice trick signal that's distinguishable in the presence of calcium chloride buffer, whereas something like TSEP or EDTA are not as favorable and should probably be avoided in the screening exercise. Just to show you a result or readout you can perform either single dose or full affinity curves with Dianthus, and this is just our control sample of S-odenosyl methionine, where you can see the reference gives a signal here, and that single dose treatment produces a nice trick signal. And you can do the full affinity curve here, titrating the amount of SAM, and the affinity determined of 13 micromolar is in good agreement with published values. So when they performed the screen, they identified a series of hits. So as I mentioned, they screened almost 2,500 different compounds, and 89 hits were determined. We also were able to get from this, as the software automatically processes the data, also identified 9 samples that were caused by aggregation. There was some autofluorescence that was observed, and of course the non-binders that came from this experiment. As I mentioned, this is just showing one particular hit from that experiment where you can first do a single dose screen. So it took them about 6 hours to screen under single dose duplicate samples, 2,500 different fragments in single dose, as shown here by these examples here. And here's the trick readout. And then another approximately 1.5 hours to go back and screen those 88 hits with full affinity curves. And this is one example here of one of the hits that came out of that screen. So in total, they were able to use less than 20 micrograms of target protein, and the KDs that they obtained were in the high micromolar to millimolar range, which was expected from the fragment library they were testing. So in summary, they were able to efficiently screen a broad chemical space while consuming very small amount of sample. They generated a total of 5,784 data points within this 8-hour period, or 1 day you could do all this screening. It only used 20 micrograms of G9A target protein for both the single dose and the full affinity screening results. 88 hits were identified that ranged from approximately 100 to 350 Daltons in size, and the molecular complexity of these hits ranged between 90 to 400. So really, Dianthus can efficiently address the screening challenges that researchers face really in doing drug discovery. Most involved is that Dianthus can help with hit identification, doing fragment-based and small molecule single-dose screening. You can further go on to do hit validation by doing full affinity curves with Dianthus and further validate and rank the affinity of your positive hits, and then transfer those to lead optimization, where you can now further characterize, doing a deeper characterization basically, and working to optimize the affinity of those lead candidates that will go on to further drug development and hopefully become new and novel biotherapeutics. So consider making Dianthus your trusted collaborator for affinity screening. You can swiftly analyze 384 data points in 30 minutes. You don't have to worry about regular maintenance as it runs with no fluidics, so there's really no downtime when you're running your Dianthus system. You can measure a wide range of affinities from picomolar to millimolar, all within the same experimental screen. It consumes only a small amount of both your target material as well as compound material, so you can conserve your precious samples. And all characterizations are done in solution, and there's no immobilization steps that are required to run a Dianthus assay. So at Nanotemper, we're focused on helping you, the researchers, find solutions to better characterize your proteins. Today I spoke to you about Dianthus, which is our high-throughput screening solution for measuring binding affinities, but we offer a complete line of tools and products to help you characterize your proteins. This includes Prometheus for precise characterization of protein stability, Tycho, which is our everyday solution for a quick check of protein quality, and Monolith, our flagship system for measuring molecular interactions. So if you have any questions, please feel free to reach out to me directly at my email address that's shown here, or visit our website at nanotempertech.com. Thanks again for your time and attention, and I hope to hear from you soon. Take care.